Hey guys, welcome back to StudyWizards. If you're in grade 11 biology and meiosis is giving you a headache, don't worry. I can hopefully help. Today I'm going to explain what meiosis is, how it works, and why it's super important, especially when it comes to genetics and making sure each of us is totally unique. First, you have to understand that there's a difference between mitosis and meiosis. Though there are a lot of similarities, for example, the names of the phases, for example, prophase, metaphase, anaphase, telophase, etc., and a lot of things that's going on in each phase, you have to understand that meiosis is a reduction division, meaning that we're trying to reduce the number of chromosomes, and essentially meiosis is done to help each gamete, which is the product of meiosis, to have the same number of chromosomes in each of them. So in order to do that, we have to do 2 rounds of meiosis to ensure that reduction is happening. That's the key difference, one of the key differences in meiosis. But also mitosis is duplicating cells. Meiosis is reducing the number of chromosomes, right? And we're producing 4 gametes. Gametes are our sex cells, aka our sperm and our egg cells, accordingly. So, again, you have to remember that every human body has 46 chromosomes. That's 23 pairs. These are called diploid cells because they have two sets of chromosomes, one from mom and one from dad. But gametes, sperm and egg cells, can't have 46. If they did, when they combine during fertilization, we'd end up with 92 chromosomes, which we don't want because then it could continue to grow and grow and grow, as it would be 92 plus 92 and so on and so forth. And there would be like a lot of genetic problems most likely. But we know that in our case, we each, as human beings, we have 46 chromosomes normally. And to maintain it, meiosis helps. And something that might get confusing further on in this video, and if your teacher does it, they do it. If they don't, it's okay. But we usually nowadays, we address chromatids as now chromosomes. So what we used to be called a chromatid can actually be called a chromosome. So that's something that got me a lot confused in the beginning because it was like, how can a chromatid be a chromosome? But it's just the naming. So I will still reference both of them as chromosome and chromatid just for your reference, for your understanding. But just know that 2 chromatids can be called a chromosome, but also 1 chromatid can also be called a chromosome. But essentially, just a little overview of meiosis. We want to start with 92 chromosomes, right, from the 2 parents, then make our way all the way down to 23 chromosomes, aka 23 chromatids in each gamete, right? We go from our 46 pairs, then we split up into 23, if you get it. The 46 splits up to 23 pairs, aka 23 chromosomes with 2 chromatids. Then we line them up in the middle, split, we get 23 chromatids, aka chromosomes in each gamete. It all sounds confusing. Trust me, it is confusing, but as soon as you understand it gets really simple. Now, here's just like an image of what the diploid versus haploid cells look like. But essentially, meiosis just turns diploid cells into haploid cells, which is, haploid is just a cell with 1 set of chromosomes. That's 23 for us humans. So the main goal of meiosis is to cut the chromosome number in half, create 4 gametes, and make sure each gamete is genetically unique. And it does this through 2 rounds of division, meiosis 1 and meiosis 2. Now, let's start with meiosis 1, also called the reduction division, because it's where the chromosomes are halved. So prophase, right, the names are very similar to mitosis, but a lot of different things happen. Now, prophase is big because the homologous chromosomes, aka the pairs of chromosomes that have very similar genetic information, line up together, and they form something called tetrads. And tetrads are basically 4 chromatids, aka chromosomes, so 2 pairs of chromosomes together in the homologous chromosome pairings then they swap genetic materials in a process called crossing over. Now, crossing over, think of it as a high five. Go imagine you're high-fiving your friend and you're literally switching your hands with them, each other. So it's the same thing. But in this case, within their chromosomes, within the tetrads, within the pairs, they're switching. And it's randomly, and it could be anywhere throughout the genetic information in that chromosome. So that's a huge reason why there's a lot of genetic variety. And it's literally mixing up mom and dad's genes. So think of the blue one as dad and think of the red one as mom. Look at the third image where anaphase is, and you can really see that swapping, and you can really have a good idea of what it may look like. So now metaphase, the tetrads line up randomly along the middle of the cell, and this is another reason why we're so genetically different, is because of independent assortment. The way that they line up really can alter how the gamete might look and how you look because of how they're lined up. Look at metaphase 1. You can see how it's blue, blue, red, and on the opposite side it's red, red, blue. It could have been red, 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 or blue, 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 but because of the independent assortment, it's blue, blue, red, and red, red, blue, and it could be different. Remember, this is just a snapshot. There's 23 of these pairs lined up, and that could cause so much genetic variation, even though the crossing over is already doing a little bit of it, but the metaphase is also doing that as well, the independent assortment in it. Now, anaphase 1, the chromosome pairs are pulled apart. One whole chromosome from each pair goes to each side, and that's where I was saying that the pairs get split up. But they're still duplicated in their little pairs. Then telophase 1 and cytokinesis, the cell splits into 2, each with 23 chromosomes, aka the 23 chromosomes with the 2 of the chromatids. Still diploid looking, but technically I'd say they're getting ready to become haploid, and I'd say personally as soon as they split, the cytokinesis happens, I'd say they're haploid, but of course ask your teacher what they think. Now moving on to meiosis 2. So meiosis 2, it looks a lot like mitosis, but there's a few differences, of course. Like, for example, prophase 2, the similarities are like the simple things like the new spindles form on both cells in prophase 2. No duplication in this sense because we're already reducing. We're trying to reduce it even further. Metaphase 2, this time the chromosomes line up in a single file, no pairs, because of the fact that the pairs are already split up into 2 different cells already. So they line up in a single file, and now they're split up according to their chromatids, aka, like I said, it might be confusing, but chromosomes at the same time. The chromatids are, aka chromosomes, are pulled apart, and there we go. The 4 haploid gametes are produced as soon as telophase and cytokinesis are completed. Each has 23 chromosomes, aka chromatids, and they're all different. So, the real question is, why is meiosis so important? Well, because of 2 major reasons. It makes sure we have the right number of chromosomes. And it also makes sure that every gamete and every person is unique. And thanks to crossing over and independent assortment, no sperm or egg cell is exactly the same, even from the same parent. And that's why, even look at your sibling. Even though you have the same parent, you guys look very similar in certain parts, but also very different at the same time. And that's thanks to the crossing over and independent assortment. And there you go. That's meiosis. The reason why every human being is one of a kind. So I hope you guys enjoyed this video, and I hope I helped. I will leave some links to some videos you may want to watch after in the description below. I hope you guys enjoyed, and I hope you guys learned something. If you guys have any questions, leave them down below. All right, bye. Thanks for watching.